out is the very raw data we get from MaxQuant. Like, what are we actually starting with? And then I'll kind of walk through some things. So this is one of the raw output files from MaxQuant. Like I said, it's got these protein group column ID. This is probably going to be very confusing because it just is. You've got multiple UniProt accession IDs listed in each of these rows. This is taking the peptides, you're summing the intensities for all the peptides to the protein level. So what we're looking at in the rows are all the proteins that were identified, and then all the columns is all the other information associated with that particular protein ID. The FASTA header is the one that's going to give you all of the information associated with that FASTA column header information. And then the database will look for certain patterns in that header to try to parse it out to the protein name, the gene name. Sometimes they'll give you the gene symbol name, depending on how you set the parsing rules. So I like to have the FASTA header there in case something didn't parse out correctly, then we at least have all of the information that was provided in the FASTA header of that database search. Another thing, if you have a project where somebody has added a tag sequence to a particular protein, we can actually add that protein sequence into the FASTA database as a new protein, and then we can identify it in our database, in our search results. But you have to know that information up front. So you can customize those databases depending on if you have extra proteins or modified, like mutated proteins in your sequence that you know about. We can make those changes before we do the database search, and then we'll have it in our data at the end. So we have done that for several projects. The protein ID, this is where we have that issue with the shared peptides. So what it will do is sometimes you'll have a cluster of proteins that are identified, and the protein that has the most evidence for that identification is listed first, and then the following proteins. So this particular one has this MAG-OHB versus MAG-OH. So it's the same type protein, the Nashi homolog 2 versus just the homolog. So they're going to be a similar protein, but it can't necessarily distinguish between those two based on shared peptide information. So you can kind of run into that issue, and that becomes more of a problem when you're trying to do pathway type analyses later, which I'm hoping that we'll have a nice discussion on annotation and pathway stuff tomorrow. So yeah, we got that to look forward to. Then, so what I do for the analysis is actually truncate this to that first ID that is listed. So when we're doing our analyses and you have the UniProt accession numbers, it's going to be pulling that very first one that has the most confidence for the identification into a nicer data frame to do all of our processing. So that is one thing that I am using. And you can kind of see that here, the majority of protein IDs. Some of them have a different column that's the leading protein. Just again, kind of depends on the parsing of stuff, but I'm not seeing that one in this particular output. Then there's going to be there's a lot of information in here that I don't actually utilize. So the number of proteins, so we had 4 in this particular group that are kind of shared. The number of peptides can be important because if you have a lot of peptides identified, you got more confidence in the identification for the summing, but that can be dependent on also the size of the protein. So this one we had 14 peptides that are related to that particular RAS protein. Then a lot of other count, score, metrics, sequence coverage, how much of the sequence did we actually identify, percentage coverage. That's sometimes helpful to know that, like, we just identified one peptide for this protein. So depending on how strong of an identification that protein is, you're basing all your results on that one peptide instead of a higher percentage of the full protein sequence coverage. So those are some things that you can look into when you're really trying to investigate the analysis at a deeper level. Scores, I have a slide deck that has some of the MaxQuant definitions, so I'll put that into the Slack, and then that way you'll have the definitions of these different scores and stuff, and you'll have those notations. What I wanted to point out here is the reporter intensity corrected channels. This is the columns that we were pulling out from the Proteonorm app to do the normalization on. So these are going to be those TMT reporter ions that are corrected for their errors in the shifting with the TMT lot. So you have the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, because those are going to be the 11 channels from the TMT multiplex. And the only way you know what 1 is versus 11 is by that lot number description in the Thermo Fisher form. They have a PDF that says it's the 126 and 127 in 127C. All of those different mixings are in the order of 1 to 11 in the output of MaxQuant. It doesn't tell you which actual isobaric label it is, it just orders them 1 to 11. So you want to be careful when you're, the core helps take care of that. They do the sample labeling, and we make sure that we have the right sample with the right number so that we can make sure that your sample names match up to the right information. So there's a process in place for keeping up with all of that information. And then if you'll see here, I actually have this TMT1 tag versus the TMT2 tag. That's an experimental design label that I gave the samples in the database search of MaxQuant. You can set the label of your samples, what you want them to be. So I just said TMT1 for the first batch, 2 for the second batch. And then we make a targets file, kind of like you do with genomic data. We just make the sample targets file. This one's a little different structure. Usually I have like the reporter ions in here, and then which replicate it is with the information about it, sample names, group names, batching. If they're paired samples, we can add a column here for paired analyses. So all of those statistical questions that we want to ask up front, we're setting in this sample group targets type file. That way, when we process our code, we have it set up to where it can automatically detect if we need to do a batch correction or a paired analysis or not in the statistical design. So we try to help ourselves out and be able to put the variables within this one file and not having to change the code every time we run a project. Let's see what else to in here. So these are going to be your raw values. And you can already see there's this missing set of intensities in all of those columns. So that particular protein had just enough of a MS spectra to be identified, but not enough for a quantitation. So we would actually filter that out. If there's a 0 across all samples, that gets filtered out of the analysis. I have depending on projects, I can filter to where 2/3 of the replicate have to have a value in one of the groups. You can set different levels of filtering to kind of get rid of some of those proteins that don't have that much, you don't have replicate information on. So there are some levels of filtering that you could actually do prior to doing any normalization or statistical analyses. And that can be kind of project dependent. So a lot of times if I'm kind of questioning what to filter, I run it with all of them. And then you can look at those Limma plots and evaluate it a little bit more. And then you can do some filtering and rerun it with removing some of those missing data. But sometimes those low abundant molecules, proteins will pop out if you don't filter them out, and they can be biologically interesting. And so I try to err on the side of not throwing too much out because it is discovery phase. But if you're just interested in the top abundant changes, then you might as well just filter it out. Depends on what your goal is and what you're interested in. But there's ways of handling all that. So these, if you'll notice, they don't have that corrected. So that's going to be the original intensity without that reporter ion error correction information. So it does matter which columns you pull out to do the analyses. And then we have the count data. And the count data is the number of MS spectra that we have for each of those proteins. So yeah, zeros is not really there. So that should get removed.
Stephanie, can I just ask a question there? So the one that has 34, that would have maybe 34 different peptides that all have those tags.
Yeah, MS2 spectra.
That corresponds back to that particular protein. Well, the other one maybe just has one. We only found one peptide, so therefore it was only one tag.
Right.
Does that seem okay? Thank you.
Yeah, and that's something if you do like a label-free project with count data, like we do with IP experiments. So you would actually take one that's a pull down that you're trying to find all the proteins associated with a particular antibody versus a background control. And so for count data, we actually set a filter that it needs to have 8 spectral counts or more to be out of that kind of contaminant type level. So you can have some filtering at the count level with label-free type data as well. All sorts of things you can do with data. That's what makes it fun, right? And challenging. So at the end of this big file, we have this reverse and potential contaminant column. These are the ones that are flagging the reverse sequence information. So this has that reverse at the beginning of that label. That means it was matching the reverse sequence of that protein and should be removed. This is how you're calculating a target to decoy ratio for your false discovery identification. And we're trying to set that threshold to 1% for identification error. So that's the reverse. And then we have one that's for contaminants. And the contaminant one, these are the proteins, that's a reverse and a contaminant. So that's even way worse. But the contaminants are the ones associated with the contaminants database. So if I find another one, not that many in that one. These are my shortcuts. Yeah, and this one, the contaminant's actually that last one in there, which is kind of interesting. I've seen some goofy things with MaxQuant output files. Sometimes they're not all consistent, like this right here. And this is like a programmer's nightmare because it's never a consistent pattern. And so you have to kind of find these empty columns in here where it's just a bunch of gibberish, and they can kind of mess up your data frame. So when we're pulling out information out of this, you may only pull the first few rows and not all of the data, just due to having these kind of extra little miscellaneous identifiers in here. So I have seen that quite a bit too. So one way to get around that is actually to sort this by this ID column first, and then all of those will be at the end, and you can get rid of it and stuff. But starting with just MaxQuant output is really painful. It's kind of a painful learning process. So from the, what we would do with the proteomics core, once you submit your samples, it goes through the mass spec, then the bioinformatics team, we take over and process the data. We do the normalization of all the data sets, and then provide the differential expression analysis at the end. So any questions about this file, I'm gonna close it.
I have another question. Do you consider any, like when we talk about RNA-seq data and we're looking at counts, like anything under 10, we kind of in the core think of it as noise and don't really trust these. If we see 2, 2, 2, 2, 2, like we'll get rid of that gene, right?
Right.
In terms of reporter ions, is there a level that you just consider semi-noise if you see it all the way across? Let's say it's 1,000 across all of them. It's like, well, this is probably not a true abundance measure. Do you have a level that you look at or not really?
We don't have that yet for these, for the intensity level, but I kind of utilize the MD plot to kind of look for those. And then we can set a threshold with that plot if we need to remove some of those lower abundant ones. But yeah, there's not really a set threshold for filtering on that level yet.
Great, thank you.
But there should be, I think. It's just kind of that abundance issue.
Yeah, we don't have one either. I don't really filter on low intensity, so I was just wondering if you guys do. Yeah,
no, not yet. Okay, we already looked at proteonorm. I've got some QC figures that we do. It's just the clustering and PCA, so I'm not really gonna go over that anymore. But our results file, this is what we would actually provide as the results. So we have our significant list at 2 different levels of significance, whether it's with the unadjusted p-value or the adjusted p-value. And then we have the full result table, and then static images of the plots, as well as the interactive ones. So one thing with these is the interactive plots, you'll have to, when we load these into a box folder, we try to zip them to save space. And in order for them to open up, you have to extract it and unzip the files. So then once you have these files, this HTML report is actually pulling the data out of these other 2 style sheet folders. So that's why it's got to be unzipped. And then you can open it into a Chrome web browser. And now we have our interactive plots. So this is just trying to take all of that output and put it into an interactive platform so you can quickly look and see what protein it is, what p-values are, and then connect it to the normalized intensity values. And because a while ago, this is really a tool that I use a lot to kind of look for things like this where you have a missing value or not. One thing on this analysis, we're displaying the missing values as a 0 in this plot, but it doesn't mean it was a 0 in the analysis. There's one thing that you have to watch out for with the Limma when you're doing an LM-fit model design. If you have your missing values set as a NA instead of a 0, then any protein that has the NA is omitted from the statistical analysis. If it's a 0, it's included and will impact your fold change. So it depends on how you set those missing values before you do the statistics, kind of how it's going to impact your fold change information. So I have found that kind of little caveat. Sometimes with IP experiments, I kind of have included it as a 0 because you're really, it's more of a present absence kind of analysis. You're looking for what was in my background, subtracting those proteins that bound to a background beads versus what comes down in my actual antibody target. So kind of have to think about the analysis a little bit differently with IP experiments. But yeah, we would provide all of this data for the end results, zip all that up, give it to the end user along with the scaffold file, all of the raw MaxQuant output so you can do any type of analyses. And ultimately what we'd like to be able to do is to partner with the idea states. So whenever a user comes from a particular state, we can connect them to one of the other bioinformaticians that can help with the downstream processing of pathway analyses and any of the extra analyses that they want to get to a publication. So I'm really hoping we can utilize this network to kind of provide that resource to help one another out and help our end users get to publications. So would love to have that happen because there's a lot of data to look at. Alrighty, so all of those results would be sent to the end user. we do try to make, and I don't think I have it in that one. I think I made it in this other example. Let me look at it. And it may be actually in this one. Like I said, I got a lot of stuff. So trying to make a simple kind of Excel results file at the end of the project that's going to contain most of the data needed for making heat maps, any other statistic, like any other plots that people need for publication, get to their significant list. So we tend to kind of make like a more Excel-friendly version of all the output that will have the headers up here, what these columns represent, which I haven't done on this one. But these are each of the individual samples from this TMT experiment and their normalized intensity values. And you can see where the NAs are. And then next to those are each of the different comparisons that we did for the statistical comparisons, along with the output from Limma. So one thing to note on the Limma analysis, so we have the fold change information is log2 scale. It comes out as just log FC, but it's a log2 scale fold change information, followed by the confidence interval, the left and right confidence intervals of that fold change information. And then you have the average expression versus the T statistic, unadjusted p-value, and the adjusted p-value, along with the B statistic, which is the log odds ratio. The added columns that we've included also include this sig p-value, our significant FDR. And what these are flagging are the ones that are significant by both a p-value and a fold change. So you can quickly kind of filter through here to get to my significant list at this unadjusted p-value level in the samples. So it's just a way of filtering through the data with two different metrics of significance, a more stringent threshold. If with the confidence intervals, if that fold change happens to have a confidence interval that is on the same direction, either both positive or both negative, then you're going to have a significant unadjusted p-value. Because this unadjusted p-value here is taking into account the standard error of the fold change information. So it already does kind of calculate some of the fold change information within the unadjusted p-value quantitation. So when I'm trying to do like a pathway type analysis, I'll tend to expand out the significance to include anything with an unadjusted p-value significant and not the more stringent cutoff, if that makes sense. Because for the pathway, I'm trying to kind of include more molecules in order to do a better pathway type analysis. But it is interesting to kind of look at your changes of fold change information compared to the confidence intervals. So if like this particular one, it goes across a 0, you have a negative, and you have a positive value. So that one, your unadjusted p-value is actually not significant. And it's reflective of that confidence interval. So, but we do try to kind of give people a list of here's your top significant proteins that we have confidence in, and kind of a more stringent list to start with. Does anybody have questions about that? So when we go through this full analysis, we'll run the ProteinGroups, we'll kind of give like the metrics of what the methods look like. We'll tell you which ones we used and provide all of those differential abundance results. And then from there is where the real fun begins, and people have to, that's where you have to dig into the biology and figure out what does this mean? Okay, yeah, these are my definitions. I'll send to y'all in the Slack. We got that, we got that. I think we are about ready for our discussion section.
Okay.